Okay, friends, today our topic is oxidative decarboxylation of pyruvate. We have studied the glycolysis. From glycolysis, the glucose breakdown occur and pyruvate molecules are formed. Two pyruvate molecules are formed. And this process was occurring inside the cytosol. Once glycolysis occurs, then pyruvate enters the mitochondria. Because pyruvate is a polar molecule, it cannot pass through the mitochondrial membrane, which is the lipid bilayer membrane, by passive diffusion. That's why it requires a transporter. That transporter is called pyruvate translocase, and that is actually a co-transporter of pyruvate and proton. It will transport one pyruvate molecule along with proton inside the mitochondria, and during this process one ATP is utilized. Once pyruvate enters the mitochondrial matrix, this part is called mitochondrial matrix. This is the outer membrane, this is the inner membrane. The inner membrane contains finger-like projections. These finger-like projections are called cristae. Electron transport chain will occur here, but the conversion of pyruvate to acetyl coA and the Krebs cycle will occur inside the mitochondrial matrix. So how pyruvate is converted into acetyl coA? This process by which pyruvate is converted into acetyl coA, that process is called oxidative decarboxylation of pyruvate. So pyruvate is converted into acetyl coA by the enzyme pyruvate dehydrogenase complex. And this pyruvate dehydrogenase complex, as the name indicates, it is a complex, means it is a collection of many enzymes. It is not a single enzyme. It consists of three functional enzymes and two regulatory enzymes. The three functional enzymes are phosphate dehydrogenase or pyruvate dehydrogenase or pyruvate decarboxylase. This is the first enzyme, and that's why it is also called as E1. The E2 or second enzyme is dihydrolipoyl transacetylase. E3 is the dihydrolipoyl dehydrogenase. So these three enzymes are the functional enzymes of pyruvate dehydrogenase complex, while two regulatory enzymes are pyruvate dehydrogenase phosphatase and pyruvate dehydrogenase kinase, and these both enzymes are regulating the function of this E1 of the first enzyme, pyruvate dehydrogenase or pyruvate decarboxylase. So now we will study how uh, it also require this enzyme also require many cofactors, actually five cofactors. The first enzyme E1 or pyruvate dehydrogenase, it requires thymine pyrophosphate, while the second enzyme dihydrolipoyl transacetylase or E2 require coenzyme A and lipoamide, while the E3 or dihydrolipoyl dehydrogenase require fulvin adenine dinucleotide and nicotinamide adenine dinucleotide. So the process oxidative decarboxylation of pyruvate is, it is the net reaction in which pyruvate, look, see, pyruvate is a polar molecule. It contains negative charge at the carboxylate group. It is the carbonyl group and it is the methyl group of pyruvate. So pyruvate is a 3 carbon compound, while acetyl CoA is a 2 carbon compound. Because during the conversion of pyruvate into acetyl CoA, one carbon is removed as carbon dioxide, and during this process one NADH is formed. So the net reaction is pyruvate is converted into acetyl CoA, one carbon is released as carbon dioxide, and one NADH is produced from NAD during this decarboxylation process. So this was the net reaction, but actually this whole process occurs in 5 steps. We will study that in detail now. Conversion of pyruvate into acetyl CoA. So the first step is decarboxylation step, means removal of carbon dioxide. It is the pyruvate. In first step, pyruvate combines with thymine pyrophosphate. The enzyme is pyruvate dehydrogenase or pyruvate decarboxylase or E1. So the carbon is removed as carbon dioxide, while the uh, that this carbon dioxide is actually this carboxylate group, and at the place of this carboxylate group now TPP will bind. Thymine pyrophosphate will bind at the place of carboxylate group. So the compound formed will be called as hydroxyethyl TPP. Because it contains two carbon atoms, so it is an ethyl. Hydroxyethyl TPP is formed in the first step and carbon is removed as carbon dioxide. In the next step, the hydroxyethyl TPP will combine with lipoamide, and the enzyme working here is dihydrolipoyl transacetylase. It is also known as E2, and the compound formed will be acetyl lipoamide. So in this step, two things occur. One is the removal of hydroxyl group, one is the removal of thymine pyrophosphate, other is replacement of hydroxyl group by carbonyl group. So when hydroxyl group is removed by carbonyl group, then this compound CH3CO, this is called acetyl, and at the place of thymine pyrophosphate lipoamide is attached. So it is now called acetyl lipoamide, and thymine pyrophosphate is now free. It will be reused for the next pyruvate molecule. This first step, decarboxylation step, is actually a rate limiting step. Rate limiting step is the decarboxylation step. The second step is the reduction step, and the third, fourth, and fifth steps are the oxidation steps. In the third step, acetyl lipoamide is converted into reduced lipoamide and acetyl coA. Actually, in this step, acetyl lipoamide combine with coenzyme A. The enzyme working is the same which was working in the second step, dihydrolipoyl transacetylase. So, at the place of lipoamide, coenzyme A will combine, will attach, and the lipoamide will be removed, and it will be also reduced because hydrogen is added to lipoamide. So this step was the oxidation step because here oxidation of acetyl lipoamide occur. Then in the fourth step, lipoamide, which is in reduced form, it will react with FAD, fulvin adenine dinucleotide. The enzyme working here is E3 dihydrolipoyl dehydrogenase, and lipoamide will be oxidized because the hydrogen which was present in lipoamide will be added to FAD, and FAD will be reduced, but lipoamide will be oxidized in this step, and FAD is reduced because hydrogens are added to it. In the last step, FADH2 will give its proton or hydrogen ion to NAD itself. It will be oxidized, and NAD will be reduced, will be converted into NADH plus H, and FAD is now oxidized. So one NADH is obtained by this whole reaction, and one is acetyl CoA. So the net products of oxidative decarboxylation of pyruvate is formation of acetyl CoA, and one molecule of NADH is also produced during this reaction, and one carbon is removed as carbon dioxide. So now we will study about the regulation of pyruvate dehydrogenase complex enzyme. I told you the only first enzyme, pyruvate dehydrogenase or pyruvate decarboxylase, is regulated, while other enzymes are not regulated. And the regulator of these enzymes are two enzymes. So those two enzymes are, one is pyruvate dehydrogenase phosphatase, other is pyruvate dehydrogenase kinase. Pyruvate dehydrogenase phosphatase. Phosphatase, I told you in the previous lecture that phosphatases are those enzymes which remove phosphate from any substrate or from any enzyme. So it is an enzyme which will remove phosphate from the pyruvate dehydrogenase enzyme. Don't confuse pyruvate dehydrogenase phosphatase enzyme with the pyruvate dehydrogenase enzyme. Pyruvate dehydrogenase enzyme is a functional enzyme which is regulated by this enzyme, pyruvate dehydrogenase phosphatase. This enzyme will remove phosphate from the pyruvate dehydrogenase enzyme and will activate that enzyme, while the pyruvate dehydrogenase kinase enzyme will add phosphate to pyruvate dehydrogenase enzyme and will inactivate it. So pyruvate dehydrogenase enzyme is inactive in phosphoform while it is active in dephosphoform. So pyruvate dehydrogenase kinase is stimulated by acetyl coA, ATP, and NADH. So these three compounds will stimulate this enzyme, and as a result, this enzyme will causes phosphorylation of phosphate pyruvate dehydrogenase. As a result, pyruvate dehydrogenase will be in inactive form, while the pyruvate enzyme while the pyruvate compound is inhibiting this enzyme. As a result, this enzyme cannot phosphorylate the pyruvate dehydrogenase enzyme. As a result, pyruvate dehydrogenase enzyme will be active and will convert this pyruvate into acetyl CoA. So this was the regulation. The next is we will study about some vitamins which are required for the pyruvate dehydrogenase complex or for the formation of its cofactors. Vitamin B1, vitamin B2, vitamin B3 and vitamin B5. These are the four vitamin B. These are required for the cofactors for the formation of cofactors of pyruvate dehydrogenase complex enzyme. Vitamin B1 or thiamine is the required for the formation of thiamine pyrophosphate. Thiamine pyrophosphate is a derivative of vitamin B1. Vitamin B2 or riboflavin is required for the formation of FAD, while the vitamin B3 or niacin is required for the formation of NAD, while pantothenic acid or vitamin B5 is required for the formation of coenzyme A. This thiamine pyrophosphate was the cofactor for E1. FAD and NAD were the cofactors for E3 dihydrolipyl dehydrogenase, while CoA was the cofactor for E2 dihydrolipyl transacetylase. So this was our topic oxidative decarboxylation of pyruvate. It is completed. In next in lecture we will study about the Krebs cycle.